Good afternoon, everybody. I'm Bill O'Hearn. I'm Development Director and Fellow at Clare College, and I'm going to be emceeing this session. We're just letting our participants in, and I'm going to let that number settle down, and then I will introduce Lynn. So bear with us. I'll keep silent for about 10 seconds and just make sure this is settled down, and then we'll begin. Okay, well, good afternoon, everybody. It's a real pleasure to welcome Lynn here today to speak with us. Lynn came to Clare in 1984 as an undergraduate doing NatSci, and then stayed on to do her PhD with Professor Ron Lasky in the Department of Zoology and the Wellcome CRC Center, now the Gurdon Research Institute. During her PhD, she was a fellow student with Anna Philpott. Many of you will know Anna from Clare, fellow and director of biological NatSci. Lynn enjoyed everything Clare had to offer. She was a college table football champion in pairs. Twice she was a runner and spent too much time on the river rowing and coaching, and took part in a large number of college sports, as well as being awarded several half blues in university athletics. She also served on the Clare Mayball Committee and was responsible for catering, a lesson in logistics that she's found useful to this day in both her scientific career and also in feeding a hungry family during this lockdown. After Cambridge, she took up a postdoctoral position in cancer research in Dundee with Professor David Lane, where she was subsequently awarded a Royal Society of Edinburgh Personal Research Fellowship. She then moved to Oxford in 1996, has been there ever since, running a lab studying the molecular biology of aging. She also teaches biochemistry to biochemists and medics in her role as Associate Professor of Biochemistry and the George Moody Fellow and tutor in biochemistry at Oriel College, Oxford's sister college to Clare. Last year, she was appointed to the Strategic Advisory Board of the All-Parliamentary Group on Longevity and is the founding member of Scientists of Health and Economic Resilience, a group trying to ensure health resilience is at the core of rebuilding the UK as we start to emerge from COVID-19. She's just recently been instrumental in securing a major grant for Oriel for a long-term investment in research and study in this area for graduate students, which is really a magnificent thing and for which she's to be congratulated. And it's incredibly important work that she's doing and very interesting to us now. So I'm going to turn things over to Lynn. And at the end, we're going to take question and answers. And we're going to do that by you typing your questions into the chat function at the bottom of your screen. So type in your question, hit return, and at the end, we'll go through those. So feel free to put those in, but we'll do those at the end. Lynn, over to you, and thank you for being with us today.
Thank you very much, Bill. I'm going to just start sharing my screen now, so bear with me while I do that. And hopefully you can all see this now. Okay, so as Bill's very kindly said, I work on aging. I've been doing it for quite some time. So aging in Oxford since 1996, quite a long time to age. And what we're interested in is not just how we age, but how we age well and how we can actually build health resilience. So the way that people stay healthy throughout the life course. So what I'm going to do in this talk is start off by trying to define what we mean by aging, and then look at cellular changes that occur as we age. Address a few strategies in the field, not just my own research, but across the field of geroscience, the science of aging, to look at how we intervene in the aging process. And then very briefly at the end, obviously it's rather topical, looking at how we can try to build resilience into health in a time of COVID. Okay, so everybody knows what aging is. You are pretty able to judge how old somebody is by looking at them. But what does it actually mean in terms of a process? Well, it's very dire. Aging is the progressive loss of bodily function over time that increases the probability of death. That's the best biological definition we've got. It's a bit bleak, but unfortunately, that's how it is. But what we're interested in is improving health in aging, not getting rid of death, but improving the health. And this is really important because what we term as healthy aging actually isn't particularly healthy when you look at it. So if you look at a 30-year-old man and assess everything that happens in his body is sort of 100% perfect. And then you look at what happens just 45 years later. So when he's healthy in age 75, his brain has shrunk to almost half. That's quite massive shrinkage of the brain. Hand grip strength, again, down to half. And hand grip strength, surprisingly, is one of the best predictors of all-cause mortality. So losing that physical strength is bad news. You may notice that older people put a lot of salt on their food. It's because it doesn't taste very good. You lose up to 2/3 of your taste buds by the time you're 75. Which impacts on all sorts of life things like appetite, your choice of foods. But most importantly, it's the ability of the body to restore normality after it's been put out of kilter. So even things like going for a brisk walk, an older person will struggle to restore the pH of their blood after they've done that. So massively decrease ability to get back to normal after any insult or any change. And it's this loss of homeostasis, so loss of balance, that seems to be very, very important in leading to diseases of aging. Now, one thing I've got to emphasize is that chronological aging, the number of birthdays you've had, is absolutely not the same as biological aging. And there are many different factors that affect your life expectancy, affect the rate at which you age biologically. One of them you can't really do much about. It's what chromosomes you inherit, so what biological sex you are, but other genes as well contribute. So in terms of sex, women tend to live longer than men. They tend to get more diseases later in life than men, so it's a bit of a balance. But in terms of genes, there are some very, very long-lived people. So this is a photograph of Jeanne Calment. She took up smoking at 104 just to see what it was like. She lasted, well, she only smoked for 6 months, but she lasted for 122.5 years. Now, she's the longest known human on record. So she had good genes, but there are people with genetic disorders that massively shorten their lifespan. So here's a photograph of a couple of children with Hutchinson-Gilford progeria. They have just one mutation in a lamin AC gene, and it shortens their lifespan. So some die as young as 2, the median lifespan is mid-teens. So a tragic disease of premature aging caused by a single genetic mutation. Now, in the normal, these are outliers. So in the normal population, genes are thought to account for about 20% of longevity. So choose your parents wisely. Not always the easiest thing. But things that have a major impact on life expectancy are lifestyle and environment. And it is quite stark that a man in his 70s, who's reached his 70th birthday in the Gorbals in Glasgow, has a life expectancy of just 1 more year. Whereas a man in Chelsea and Westminster has an extra 15 years once he's reached the age of 70. That's average life expectancy. And this is all down to your environment and the lifestyle. So what you eat, how much exercise you take, all those lifetime exposures. And this is termed the exposome. Right, so what we also know is that older people suffer from a range of different diseases. When I mentioned healthy aging, there aren't actually many older people who don't have a chronic underlying health condition. So diseases that go up with age: cancer, heart disease, dementia, blindness, osteoporosis, sarcopenia, which is muscle weakness leading to loss of mobility, and one that very few people talk about, but it's critically important and a major cause of admission to care homes and social isolation, is incontinence. We really need to know what goes wrong and how we can fix it. And when we look at the incidence of some of these diseases, you can see that they all go up with age. Age is the single biggest risk factor for all of these. And these diseases also cluster. So generally, if you have diabetes, you have an increased risk of dementia. Excuse me, I'm going to cough. Sorry about that on the computer. So these diseases cluster together and they all show an association with age, a very, very strong association with age. So perhaps if aging is the single biggest risk factor, maybe aging itself is the cause. But what do we mean by that? That's sort of a tautological argument. So is there an underlying aging process, a biology of aging, that we can start to understand in order to understand why older people get so many different diseases? Well, we have to start at the very beginning, and I know it's a fairly mixed audience, so sorry if this is patronizing, but I find it useful, even teaching biochemistry, that to point out that we are made of individual building blocks, and those building blocks are a bit like bits of Lego. So these building blocks are cells, and our body is composed of 200 different types of cells, so different shapes, colors, sizes. And once you've got 37 trillion or so of those altogether, you can make a human. And every single one of your cells is susceptible to damage and changes that happen over time. So if you multiply that up by 37 trillion times, then you can imagine that there's quite a risk factor simply with the passage of time. Now, how do we investigate cell aging? Well, we do it in the lab, and here is a sterile tissue culture hood where one of the postdocs is growing human cells in these dishes. And this is what they look like under the microscope. So here's a flask of cells, and we can examine them. These are young skin cells looking quite nice under the microscope. So we can grow these cells in culture for long periods of time and look at what happens as they age. So what are the things that cause aging? Well, I've been talking about time. Obviously, we're all familiar that the passage of time does have a major impact on how we age and changes that happen as we age. And one of the things is that as we get older, our bodies have had to go through multiple rounds of repair and replenishing tissues. And so every cell in the body is dividing many times. And as they divide, the chromosome ends, the telomere caps at the ends of the chromosomes get shorter and shorter and shorter, and eventually trigger a process it's sensed as a DNA damage thing and triggers a DNA damage response. Now, other things that can go wrong are exposure to chemicals. And when we think of chemicals, we think of environmental pollution, and that is actually a major cause of aging. People who live in high pollution areas age faster than people who live in, if you like, the leafy suburbs. But when we mean chemicals, we don't just mean environmental. There are many chemicals generated within the cells simply by the process of being, of metabolizing, of making energy that are harmful to the cell. These accumulate and they cause damage to the important molecules within the cells. And then stress is a major driver of aging. And I don't just mean stress as in you have a work deadline, you're very anxious, the adrenaline is pumping. I mean chronic low-grade psychosocial stresses and also chronic low-grade stresses within individual cells. These all can drive the process of cell senescence. Senescent cells look very, very different from young cells. So here are young human skin cells grown in the lab, and they're very long, they're very thin, they're actually beautiful when you look at them. And these are the same cells grown for many, many generations until they're extremely old. There's over 100 young cells in this field of view on the microscope. At exactly the same magnification, there are 5 cells here. They are massive. They become hugely hypertrophic, so increasing in growth, and they accumulate a lot of junk and rubbish. So their nuclei, so the place where the DNA, the genetic blueprint is kept, becomes dysregulated, and they can have multiple different nuclei within the same cell. But importantly, particularly for skin, these cells don't seem to associate properly together. So we have gaps in these layers of cells. And of course, if you think about skin integrity, that's very, very important. You can see that older people with more fragile skin have problems with wound healing. Right, so we know that senescent cells accumulate with aging. We can make them accumulate in the lab simply by aging the cells in the lab. When we look at young versus older people, the number of senescent cells goes up with age. But that's not a very strong scientific argument that senescence is involved in aging, it's simply correlative. But there's now some more causative data, the first one being that if you remove senescent cells, you can improve health. And I'll show you some data for that in a minute. But perhaps most compellingly, injection of senescent cells into young mice causes premature aging. The mice succumb to various different diseases of aging across the board. So these cells are causative of detriment in aging. And how do they do that? Why are they so harmful? Well, they have multiple different effects or phenotypes that are harmful to the body. So this is a big blobby old cell. And the first thing it does is it fails to divide anymore. So if you have a wound, you struggle to repair because these cells cannot proliferate. They can't fill the gap. They can't undergo wound healing. So older tissues are much more susceptible to damage and poor wound healing. Senescent cells also secrete a whole pile of inflammatory factors, so they essentially create small fires around your body, and factors that can actually start to break down surrounding tissues. So they cause not only inflammation, but tissue damage. And then their gene expression patterns change quite markedly. So some genes that should be regulated quite tightly are simply left on. So if you think of the analogy of a tap being left running. And then they fail to recycle any worn out parts. Normally, young cells are very good at getting rid of damaged components. They recycle them and they use the individual building blocks. Senescent cells can't do this, and that's why they accumulate so much rubbish, and they simply clog up with the rubbish. But perhaps most importantly, they have all these detrimental aspects to them, and they don't die. They sit there in the tissue, causing harm through all these different mechanisms. And essentially, they're like zombie cells. And like zombies, they can go around and cause surrounding cells in the same tissue and also in other parts of the body to also become senescent. So it spreads throughout the body, causing even more harm and contributing to disease. Now, that's very depressing. So I wouldn't be here talking to you if there wasn't some good news. So let's look on the bright side now, the glass half full rather than the glass half empty. And the encouraging side of things is that we're starting to find ways to intervene in the cell senescence process to tackle cell senescence. Now, if we look at young cells moving towards senescence over many, many cell generations, there are multiple points at which we can intervene. One of them is simply to stop the cells getting old. So you can avoid this by restoring the ends of the chromosomes, the telomeres, using an enzyme called telomerase. So that sounds like a perfect fix. The cells stay young. The only problem with this as a therapeutic is that it's exactly the same mechanism that cancer cells use to become immortal. So the cells themselves may stay young, but you may not. You may get cancer. So as a therapeutic, I wouldn't necessarily recommend that one. Another potential route in is simply to replace any worn out cells. And there are companies in America now, Agex is one in particular, generating off-the-shelf stem cells that may be suitable for simply replenishing body cells that have failed to repair. So stem cells are looking like a very promising therapeutic. We can also now use drugs to suppress the secretions, the harmful inflammatory secretions of senescent cells. And then we can try and turn the clock back. So either we can delay the time at which cells become senescent, or we can even modify the senescent cells to make them look and behave young again. So drugs that do this are called senomodifiers. But if senescent cells are so harmful, why don't we simply just kill them outright? And that's one strategy that's being pursued in the field at the moment. So the initial studies were done in mice, and they looked incredibly promising. So here's data from a colleague, Peter de Keyser in Utrecht. This is a prematurely aged mouse that, when fed a drug that kills senescent cells, showed great improvements in health. So its fur grew back. I mean, this one's gray and losing its fur. This one, the fur grew back, the eyes got brighter. Whole body health improved. And importantly, this mouse was much more mobile. It was moving around and behaving like a young mouse. And then some other data from other labs around the world. So this is Jan van Dersen and Darren Baker, who also gave drugs that kill senescent cells to mice. The control mice had this lifespan trajectory, so the number of mice surviving over time. And given the drug, the median survival, so where 50% of the animals were still alive, was increased by 27%. So that's a really marked improvement in midlife health and lifespan. and then starting the treatment much later on. So Laura Niederhofer has shown that if you give drugs that kill senescent cells even late in life to mice, you can still extend their lifespan quite significantly. And this lifespan extension is accompanied by health extension. So across the board, if you look at the skin, the heart, all the various tissues, they all show improvements. The mice, if they get cancer, they tend to have better survival, but perhaps most importantly is their behavior. The mice become much more inquisitive, curious, they run around, they explore, they really do regain that enthusiasm for life, characteristic of younger animals. So this is all very promising. These are preclinical drugs. Now, if they're that promising, why don't we put them in humans? So obviously the next step is to do human clinical trials. And some of the first trials have used drug combinations. It's a bit of a mouthful, dasatinib and quercetin. So in the field, we just call it D and Q. Very interesting drug combination that kills senescent cells. And in phase 1 clinical trials, the drugs look like they're having efficacy in a really nasty disease called idiopathic pulmonary fibrosis, lung fibrosis. And so they're also testing in diabetic kidney disease, which is associated with a large number of senescent cells, and now recruiting in trials for Alzheimer's disease. And then a company called Unity Biotechnology are developing a slightly different type of drug modality. And they're looking, first of all, in osteoarthritis of the knee. And this is a condition that is incredibly painful and affects quality of life for so many people. But the only current treatment is surgery and having a knee replacement. So it's pretty drastic. It'd be much better if you could just take a small molecule drug that would improve things. And so their drug UBX0101 has gone through phase 1 and is now moving into phase 2 clinical trials. And they're also developing similar agents for age-related macular degeneration and diabetic retinopathy, so blindness associated with aging. So there's some very, very promising things happening in the field of killing senescent cells. But should we always kill senescent cells? And this is a big unknown question in the field. It's good in some tissues. I've shown you in mice, it's good in many tissues. But the big deal is we don't know how many senescent cells we have. Even in the skin, we don't know how many senescent cells we have. And this is because of various different environmental exposures. So here's a classic picture of a French taxi driver. They drive on the other side of the road, as you may remember in France. And this is the non-sun exposed side of the face. And the taxi driver drove with the window down under hot sunny conditions. And this sun exposed side of the face has aged much, much faster. And accumulated far more senescent cells. So we know from studies, particularly Unity Biotech have done this, that there's about 10%, 5 to 10% senescent cells in the arthritic knee joint. If you kill 5 to 10% of your cells, that's probably not a bad thing. In the skin, as you get older, 15% of the cells this side of the face may be okay, 60% of your cells senescent, and you kill all of them. Do you simply have your face melt off? And we don't know, and it's a slightly risky ask. And perhaps the most risky is we absolutely have no idea how many senescent cells there are in the brain as we age. So you can't ask a mouse if it remembers its grandmother, but perhaps in humans, removing senescent brain cells may not be a good idea. We simply don't know yet. So in my lab, we've taken on an alternative strategy. Rather than kill senescent cells, we're trying to modulate their behavior, so make them behave in a way that's not detrimental. But in order to do that, we've got to understand what makes them different from young cells. And it's blindingly obvious down the microscope. The cells behind me, you might see, look, I've got the young cells and the old cells behind me. They look different, but what makes them different at the molecular level? And one way we can do this is simply grow the young cells in culture for many, many, many divisions until they get old and extract the molecules from them throughout the aging process, and essentially play a game of molecular spot the difference. It sounds easy, it's slightly more complicated than that, but essentially we use a technique called proteomics, where we extract the bulk of the proteins out of these cells and simply compare the proteins that are present in young cells and those present in old cells, and also differences in the levels. And what we find from this is some horrible network analysis of different biochemical pathways. But one of the things that has popped out of this is something called mTOR, which is permanently switched on in senescent cells. It seems to be one of those key drivers of cell senescence. And essentially, that's one of the taps that is left on. And so we all know what happens if you leave a tap on. You flood the bathroom. So having this tap turned on is a problem. Can we modulate it? Can we turn the tap off? Or can we regulate it? And yes, we can. So there are drugs that can actually modulate mTOR activity. And the first very promising evidence came out as far long ago as 2009 now, using rapamycin in mice, feeding rapamycin to mice and showing that it extended their lifespan. So this was very exciting because the drug was fed to middle-aged mice and it still had an impact on lifespan. Now that's lifespan. Health, to me, is perhaps the more important measure. And a colleague, Arlen Richardson, did some very interesting studies in a mouse model of Alzheimer's disease, where the mice have enormous deposition of amyloid in the brain. On treatment with this drug, rapamycin, that inhibits mTOR, they showed much less amyloid in their brain and also much better blood flow to the brain, which is indicative of improved brain activity. And most importantly, these mice treated with the drug could find their way around a maze much better. They had improved cognitive function. So this is a really, really promising therapeutic for all sorts of different age-related conditions. And it's now being rolled out, not quite yet into humans, but into a major preclinical trial in dogs. So a project run by Matt Kaberlein in the States called the Antiaging project is recruiting 20,000 pet dogs, or companion animals as they're known in the States, because these animals share the same environment as their owners, and they tend to share some of the same habits. So if they overeat and they're lazy, if the owners overeat and they're lazy, they may overfeed their animals and they might not take them for walks. So there's a shared environment, and also there's quite a lot of genetic heterogeneity in the dogs, so you can find out a lot of things about that's relevant to the human population. And importantly, what they've already found in this dog aging project is that you can improve cardiac health in pet dogs simply by feeding rapamycin. So it's looking very promising in that respect. Now, rapamycin, obviously, very interesting drug. A lot of medics are not too keen on it because it has been used historically as an immune suppressant for preventing kidney rejection. The doses that are used in the aging space are much, much lower. So the side effect profiles are likely to be much milder. So we looked at rapamycin in our cell cultures to find out what was happening as cells age. So here's hyperactive mTOR leading to cellular senescent, the big fat old cells with loads of accumulated junk. If we treat them over a short period of time with this drug rapamycin, we can make the cells look younger. And we can also delay the time at which they take, delay the onset of senescence. So they have longer before they reach senescence. This generally is a consequence of them growing more slowly rather than them evading senescence entirely. Now, rapamycin is an interesting drug. It acts as a sort of molecular glue, bringing in another protein onto mTOR. So it has quite an interesting effect. And there are some newer drugs, second generation mTOR inhibitors now, that instead inhibit the mTOR enzyme. It's a kinase, protein kinase. And these drugs fit in the active site of the molecule and directly inhibit rather than the indirect inhibition of rapamycin. And what we find in our hands is that these second generation mTOR inhibitors are actually more effective than rapamycin in improving health of the cells and also actually improving their longevity. So growing cells in culture for 500 days, and let me tell you that is a pain. I couldn't get the postdocs to do it. I had to do it in the end because no one else would do such a horrible experiment. But continuous cell culture, we can keep these cells going and going and going, and you generate far more cells. They overcome that senescence barrier, But, and importantly, these drugs were originally developed as anti-cancer agents, and at no point over many years of working with them have we ever seen anything that looks like a cancerous transformation. So we think that these drugs are likely to be pretty safe. Now, the field has got to a point where we can intervene in aging. There are clinical trials ongoing with these drugs. So over the past year or so, I've been involved in the All-Party Parliamentary Group for Longevity, looking at how we can improve healthy life expectancy to try and let the government fulfill its manifesto pledge of healthy life expectancy increased by 5 years by the year 2035. And so we did a major study in this and came up with a national strategy report. And here's Matt Hancock waving it about, saying it's the new white paper for the government. And I'd like you to look at the dates. We launched this report on the 12th of February, 2020. And we sort of knew what was coming around the corner at this point, COVID. So how do we achieve healthy life expectancy of an additional 5 years in a time of COVID? Particularly since we know that older people are very, very susceptible to poor outcomes from COVID. So if we look at deaths by age and gender, people over 80 are far, far higher risk of dying from COVID than young people. Now, people with underlying health conditions also are much more susceptible to COVID. But that also correlates with age, as we've seen. So accumulation of senescent cells as we get older predisposes to multiple different diseases of aging. So health conditions correlate with age. Now, we know, therefore, that there are a number of contributing factors. Gender, men are twice as likely to have serious outcomes from COVID than women. Pre-existing health conditions, age is a major player, and as we're all aware, ethnicity is only starting to be explored in terms of its contribution to poor outcomes from COVID, but it's an incredibly important thing. And unfortunately, I couldn't plot any data up because even the death certificates did not reflect ethnicity, and quite recently. So it's a very underreported issue, and one that's critically important to understand. Okay, so we know that aging per se, you look at these curves, aging is actually the single biggest risk factor for deaths from COVID. 91% of deaths, of all hospital deaths in the UK have been from people in the higher age brackets. So why? And aging of the immune system, as well as aging of the whole body, seems to be a major player. This process of immune senescence. Now, just to take you back a little bit, I don't know how many people have an immunology background, probably not many. There are three major pillars of the immune system. The first is the epithelial barriers, so the surfaces of our body that are in contact with the outside world. And essentially you can think of them as the wall holding back the barbarian invaders. Now, if that wall becomes breached, we have a second line of defense called the innate immune system, and that is an ancient system, massively evolutionarily conserved. It's very quick, but it's not particularly specific. But in humans, we have multiple different professional innate immune cells, plus every cell in your body has the ability to respond through an interferon response. So we've got macrophages that eat the incoming invaders. We've got natural killer cells. Here's a lightsaber, they don't normally have that, that go and kill damaged cells, and then dendritic cells that carry messages to the next arm of the immune system, the third pillar, the adaptive immune system. And this is made up of lymphocytes, B cells from the bone marrow, and T lymphocytes also made in the bone marrow but educated in the thymus. And these are the cells that give a very specific tailored response to pathogens, and the B cells make antibodies and the T cells have multiple different functions. Now, the problem with aging is that all of these pillars of the immune system are compromised. So the barriers break down. You've already seen that skin cells don't look so good as we get older, and that's due to cell senescence. The innate immune cells have poor recognition of pathogens, plus they start checking out even more inflammatory factors. So this excess inflammation can cause harm. And then the adaptive immune system is also defective in older people due to thymic atrophy. So there's a bow tie organ, the thymus, that atrophies or degrades with age. So you have fewer T cells, and the T cells that you have undergo senescence. And then, as a double whammy, you fail to distinguish self from non-self, so pathogen from self, and start to attack yourself. And that leads to autoimmunity, which is very strongly associated with older age. Okay, so what can we do about that? Well, we can start to improve immunoresilience using anti-aging medicines. And some of the medicines I'll tell you about now, you may not associate as anti-aging medicines because they are in the clinic for different roles. So particularly statins, people will take to control cholesterol levels. But we now know from work in Birmingham, so Liz Sapie and Janet Lord and various people there, that statins improve the innate immune system, particularly some cells I haven't mentioned called neutrophils. And if older people admitted to hospital with community-acquired pneumonia, this is pre-COVID, by the way, are given statins at the point of admission, they have much, much better survival than patients, matched patients who are given a placebo, so non-statin. So statins are supporting the elderly immune system and improving outcomes. And similarly, patients admitted to intensive care units, critically ill patients, if they are already taking bisphosphonates for osteoporosis, they do much better. And again, bisphosphonates seem to have an impact on cell senescence in addition to the bone mineralization that they're prescribed for. So these are actually geroprotective drugs. They protect you from aging and they improve immunoresilience. And then the drugs that I've been talking about, the mTOR inhibitors, there's a drug called RTB101, which has a similar mechanism of action to the one that I've just shown you that we're working on in my lab. And mice given this drug are protected from a lethal dose of influenza. So this is with drug and without drug, and there's a very, very significant increase in survival if you treat with this geroprotector. So this has huge relevance to the times we find ourselves in. And the company developing this drug, RTB101, is Restore Bio. And my colleague Joan Manick, who's the Chief Medical Officer, has shared some phase 2 clinical data showing that treatment of older people, and these are people 85 plus frail, or people with pre-existing conditions like cardiovascular disease, are protected both in terms of the incidence and the severity of respiratory tract infections by taking the drug. And it reduces by half coronavirus infections. Now, the numbers are very small because at the time of this study, coronaviruses weren't very prevalent. But now Joan is moving into clinical trials in nursing homes in America. We've co-written some trials. We're still waiting on NIHR for a decision on funding, but we're hoping to put this into old people's care homes to try and give them immunoresilience to protect them against COVID. Now, there are multiple different drugs out there, so I don't think that there will be the panacea, the one drug to treat them all. And part of the reason for that is different parts of our body are made up of different cells, and the different cells age through different mechanisms and at different rates. But there are a few key principles that we can take away from this. There are drugs that kill senescent cells, and many of those are in clinical trials or preclinical development. There are drugs that modify senescent cells, so make them less harmful, and mTOR inhibitors and statins are pretty impressive on that front. There are drugs that enhance the immune system, and again, statins stand out there. And then there are drugs that improve the ability to recycle worn out components to promote a process called autophagy. So a few common features between these are some core drugs that are looking very, very interesting, and particularly mTOR inhibitors because they have been through phase 2b and into phase 3 clinical trials. But we have to step back a little bit and ask ourselves whether we should be doing all of this. I've had a lot of people pointing out that aging is perfectly natural, we all do it, and we shouldn't intervene. And I'm not trying to intervene in aging to prevent death. What I'm trying to do is improve health throughout the life course. And ethically, we do this anyway. We do this for infectious diseases. You don't deny an older person antibiotics when they get a bacterial infection. We treat all the way through life. non-infectious disease, cancer, all sorts of non-infectious diseases, diabetes, we treat throughout the life course. But what happens is we treat diseases, people are healthy in their youth, and then their health deteriorates as they get older. Now, if we treat individual age-related diseases, say cardiovascular disease, someone has stenting for a heart attack, they don't die, so they live longer. So we increase lifespan, but they're still susceptible to those other diseases of aging because we haven't tackled the underlying cause. Now, if instead we start to think of dealing with the process of senescence, the aging process underlying multiple different diseases, perhaps we can get close to the ideal where we maintain health throughout the lifespan and then eventually run out of steam, fall off the cliff. And that seems like an ideal scenario to me, that we live in good health for the vast majority of our lifespan. And again, ethically, cancer is a disease that's perfectly natural and normal. It's simply your own body cells gone bad. Senescence, again, is simply a process leading to multiple age-related diseases where the body cells go bad. So if we treat cancer, I can't see how there's an ethical argument that we shouldn't treat senescence. And one point that's incredibly important, although we say that lifestyle is important in aging, people should not be judged or blamed for poor health in later life. There are so many different factors that impact on health that we should treat people and not blame them for how they are. Now, just one cautionary point, the drugs I've been talking about, senolytics and senomodifying therapies, are likely to be dangerous in pregnancy and early development because senescence is actually a key process in developmental processes, so in pregnancy and child development. So don't take senolytics when you're young. But what we can all do to live longer and healthier and build health resilience is take sensible amounts of exercise. Don't overdo it, but take enough. Eat properly, and there's a strawberry here actually, not just because I love strawberries and it's that time of year, but because they have a very high concentration of the drug fisetin, that is a senolytic that actually helps improve health outcomes, at least in mice. Sleep is incredibly important, and we're only just starting to understand the biology of sleep in that context. So is optimism and friends and good company. And so I'm just going to end with thanking my friends and good company, so the people in the lab who've done a lot of the work, the funders who've supported it, and particularly Jim Mellon, whose vast generosity has set up an in perpetuity senescence and immunoresilience scholarship based at Oriel College. So I'll end there, and thank you for your patience, and I'm happy to take any questions.
Thank you, Lynn. That was magnificent. Please do add some questions. There's one here I'm going to come to in a minute. And in your last slide, you sort of dealt with the question I was going to ask, but maybe you could talk a little bit more. So when do you think these drugs will start to become more available? And in the meantime, since what we put into our bodies makes such a big difference in this area, in addition to strawberries, what else can we eat the rest of the year?
It's the classic question, and it's the classic answer. Lots of fruit and vegetables. If you're going for protein, go for lean proteins. If you're vegetarian, you have to make sure that you have all the micronutrients supplemented. It's really boring, old advice that you've heard time and time again. Loads of fresh fruit and veg. Go out in the sunshine, don't get burnt, get your vitamin D levels up. So yes, there are all the old-fashioned pieces of advice, unfortunately. In terms of drugs, probably a lot of people are already taking some geroprotectors. The statins are incredibly widely prescribed. And of course, people think they're taking them for cholesterol. In fact, most medics are prescribing them for cholesterol, but they don't realize at the same time they're supporting the immune system. So these are drugs that we're looking at that may have really wide application. There's another drug that I have mentioned called metformin, which is an anti-diabetic. And a colleague near Barzillai in America is trying to set up a major clinical trial. It's a cheap, safe drug as far as drugs are safe, being used in millions of people worldwide. And it seems to stave off multiple different diseases of aging. So NEAR is setting up a trial treating aging with metformin, recruiting a large number of people and following them until first onset of any one of a number of age-related diseases. So there are cheap generic drugs out there already that may help. And then there's some of the more tailored things. We're in the very early stages of drug development in the aging space, and 1,000,001 different companies all looking at different types of senolytics and different types of interventions. So they range from the ultra cheap, a few pence a day, to the ultra expensive. Gene therapy for telomerase, they're charging £1,000,000 to take part in a clinical trial that, in my book, might give you cancer. So I hope that's not libelous. I'm just saying that biologically you have to be extremely careful about extending your telomeres. So yeah, they range across the board.
A question from Elizabeth. She wants to know, should we all be taking statins and when?
I'm hesitant about taking anything without knowing the reason why in the biology. So everybody is aging at different biological rates. And what we need to do is find out what the senescent cell burden is, what the immunological function is. And if you're already hale and hearty, maybe you don't need to take them. There are some interventions like exercise that actually have a much bigger impact. Exercise is anti-inflammatory, which is quite interesting. So if you look at super cyclists, people in their 80s who are cycling 100 miles or so a week, they are ultra fit and healthy, and they have very low levels of inflammation. So there's things that we can do that we know work and don't have side effects. So I would say with the statins, and I'm very cautious on drugs, hold back a little bit until we have biomarkers to let you know whether you're the right person to be taking them.
Okay. So Elizabeth asked a question. I'm going to expand on it. So how do we find out how old we are?
Oh,
And the question there is, for our medical community, for our GPs, how are they going to get up to speed on this so that they can be treating us all?
Exactly. It's a massive, massive question in the field. We have a few biomarkers for senescent cells that aren't very robust. And so there's a massive push in the field now to develop biomarkers, so blood markers, even people taking photographs of faces, so using AI to try and work out how old you are from your face. And there are some strong correlates with what we know about rates of aging and photographs. One of the best measures at the moment is called grim age. And you might be able to tell from the name that it is the best predictor of all-cause mortality. And it looks at methylation on the DNA. So it's an epigenetic clock developed by a guy called Steve Horvath in the States. And it will predict your lifespan to within about 3 years accuracy on average. So you can have your grim age measured if you want to, if you're brave enough. I haven't done that one yet, but it is modifiable. That's the other good thing, that you can actually, with diet and exercise and all the good stuff, you can actually change your biological clock. And Steve has actually measured this in people who've taken some geroprotector interventions.
So you have to be a pretty informed customer to know what to ask and what to look for, though, don't you? Right.
Yes, it's not straightforward at the moment. So we don't currently have a blood test you can go to your GP and say, how old am I biologically? I mean, maybe there are companies out there developing stuff. So within a few years, it might be just something you buy a kit off Amazon, but not just yet.
Okay. So Matt asked a couple questions. Let me start with the second one.
Yes.
Do you think there is a U-shaped curve for exercise? Does stressing yourself with ultra marathons, etc., have a downside?
Yes. So Matt, absolutely. There are some studies. In fact, there's some people in Cambridge doing this work. The right amount of exercise, I think I put on my slide, sensible exercise. Not enough is bad for you, too much is bad for you, and actually you can cause harm if you do too much. So 10 hours of exercise a week seems to be a sensible amount. I mean, I'm not the best person to ask about this, because at Claire I did rather more than that. I was probably doing that much a day. You can actually cause detriment. It's not so much a U-shaped curve. You hit a plateau where more exercise does not do you any more good. So you can get improvements and keep getting improvements, and then there's a threshold beyond which there's no more improvement. And of course, the ultra exercisers, the ultra marathon runners, do run the risk of all sorts of skeletal injuries and musculoskeletal injuries as well. So I don't run ultra marathons.
This earlier question was at the start of your talk, an early slide, and you were talking about the life expectancy in Glasgow versus a nice neighborhood in London.
Chelsea and Westminster, yes.
Chelsea and Westminster. And were you saying that the life expectancy in Glasgow is 71, and in Chelsea it was 86? Or were you saying that if you get to 70, how much time do you have left?
It is actually your life expectancy at the age of 70. So it already rules out all those other impacts. So early life, well, it doesn't rule out early life influences, but anyone who's died before the age of 70 isn't contributing to that life expectancy curve. And Matt's absolutely right that there are Glaswegians who live a lot longer than that, and there's a postcode difference.
Okay.
So again, it's the Gorbals versus the leafy suburbs, and there can be a 10-year gradient across a 2-mile geographic tract of Glasgow. That's also true in Liverpool. I've got a colleague who says he walks to the hospital where he works, and he walks 10 years down a life expectancy curve to get to work. Wow. So socioeconomic deprivation is a huge driver of poor health outcomes and premature aging. So okay, that's something that should be intervened with. Not with drugs, but with actual providing better jobs, better employment, and encouraging people to live healthier lives.
A couple more questions. We've got some good ones here. Tim wants to know, what would push most people over the cliff if we deal with this aging and other postponements?
Exactly. It's an incredibly good question. There are a group of supercentenarians who've shown us what happens. So I've mentioned Nir Barzilai. He works at the Albert Einstein Institute, and he has studied a group of Ashkenazi Jewish people who are living to 105, 110, 115, essentially not showing signs of disease, and living very, very healthily until very late age. And then they go to bed and they don't wake up. They die of old age. So he's got to know the population very well, and he's been allowed to conduct postmortems, and some of the studies have suggested they simply run out of stem cells. There's nothing left. So one study, and I don't know how they counted it, said they had 2 stem cells left in the bone marrow of one of these supercentenarians. We've normally got millions, billions. So it looks like you run out of steam, which isn't a very biological answer, but it does look like it's a stem cell deficiency.
And that could probably be addressed at some point.
Exactly, which is where we come onto the side of aging that I find ethically a little bit more difficult. Yeah. If we keep replacing bits that wear out, how far do we go with this? Anyway.
So Isabel asks about chronic fatigue syndrome and ME, a dysfunctional immune system. Could statins be a potential treatment there?
I'm not sure about statins as such, because we know the molecular mechanism of those, and it seems to be that they are activating signaling through a small G protein, RAS signaling pathway, and it helps with cell motility. Now, ME may actually be an energy generation syndrome that's more linked with the mitochondria. So there are some studies now with the ME Society chronic fatigue research looking at whether dysfunctional mitochondria, the powerhouses of the cell, are contributing. Now, something that I didn't mention is mitochondria do become dysfunctional with aging. So they get bigger, they get more of them, but they're actually unable to fulfill their function of making energy. Instead, they get partway through and they generate harmful forms of oxygen called reactive oxygen species that whiz around and damage DNA and everything else. So I'm not sure about statins in that context, but the aging field is looking very seriously at chronic fatigue in terms of whether some of the therapeutics we've got might help. So no actual drugs at the moment, Isabel, but it is of major importance and major interest within the field.
Just different drugs cost more to make. Are these very expensive things to formulate and to make widely available? What's the issue on the development of these things?
Right. If you start from absolute scratch, I've just been trying to co-write a paper on drug discovery today. There's different ways you can do drug discovery. You can say, okay, I've got an important protein like mTOR and I want to make a drug that modulates mTOR. So that's called target-based drug discovery, and you can make wonderful drugs that fit perfectly and inhibit the enzyme in vitro, and they work in cells, and you put them into people and you put them into preclinical systems and they're toxic. So a lot of drugs fall by the wayside in drug development. And it makes drug development extremely costly, not because the individual drug per se might be expensive, but because you've got over 90% failure rate. So you invest a huge amount of time and money into things that simply don't pan out at the end. So you have to recoup your costs by charging for the few drugs that do work. One of the things that we're trying to do now is change some of the mindsets on drug discovery. So rather than saying there is a single one protein that is the be all and end all of a disease process, you look at the disease more in the whole. You look at the system that you're trying to modulate, and you use a drug maybe on a senescent cell, and you're looking at treating the whole of the senescence process, and you go in agnostic of what the drug target is. And that way round, with what's called phenotypic screening, you may have a higher hit rate, and the drugs may be less toxic because you've already tested them in a cell-based system. But drug discovery is costly and time-consuming. And one thing that's looking quite interesting is instead doing drug repurposing. So looking at drugs we've already got, drugs where drug companies have made them, pharmaceutical companies have made them, and they're perfectly good drugs, but they've put them on the back catalogue because they are no better than what's already on the market. So it's not that they don't work, they work fine, but they can't sell them for that particular indication. But as I said, the drug that we're looking at was developed as an anti-cancer agent, and now we're looking at it in an aging context. So repurposing of already formulated drugs where a lot of the toxicity and everything else has been done will save a lot of money, make the drugs a lot cheaper.
So John asked a great question. This is so important and interesting and potentially transformative and important to people. How do people keep up? Where does the civilian go to know what to do and what to read?
I would love to know. I don't know where the specialist goes to read about this. There's a huge body of literature coming out all the time. We had a fantastic colleague in the field, Dominic Burton, who had a Facebook page where he just pulled together absolutely everything from aging. I don't know how he found time to do anything else. He's moved out of the field, so unfortunately we lack that resource. There are some very good websites. So people with different perspectives on aging, someone called Reason, who runs a website called Fight Aging, and his perspective, he wants to fight aging full stop and cure death. But he's very, very good on keeping up with advances in the literature, so that's an excellent website. Yeah.
Okay.
So one thing I would suggest is don't necessarily try to keep up with it by everything you read in social media and the tabloid newspapers, because they vary from day to day. Aspirin will cure you of everything, and then aspirin will kill you. Be very, very careful about the take in certain platforms.
So I have one solution to propose, Lynne, which is you've been fantastic today. And if we can have you back again at Gala Day next year, hopefully in person, but I think this digital format's going to stay. And you were a brilliant digital presenter.
Thank you.
And really enjoyed this. And so to everybody who's out there watching, if you have one more question, type it in now or forever hold your peace. But if another one doesn't come, I just want you all to tell your friends, share the YouTube link, say what a great talk you enjoyed today, and please tune in for some other things today. So we're getting a few thank yous coming in, but I'm not seeing an additional question. So thank you for
breakthroughs on Alzheimer's.
Yeah,
Sorry. Okay, do you want me to take the Alzheimer's question?
Absolutely.
Okay, right. Alzheimer's is a very, very interesting study at the moment. For 30 years, we've been hung up with the amyloid hypothesis, that amyloid is actually causative of Alzheimer's. And all the drugs that clear amyloid have very, very little impact in disease modification. And then last year, a very interesting study came out suggesting that gum disease bacteria secrete proteases called gingipains, and these get into the brain and they actually cut the amyloid up more quickly, and you are much more likely to get Alzheimer's if you have gum disease. And they've come up with some drugs that inhibit the gingipains that might look quite promising. And then some studies that have been around in the literature for years. Ruth Itzhaki has suggested that there's viral infection in the brain that might be triggering Alzheimer's. And then very recently that's been picked up on again, that in cell culture, you can trigger an Alzheimer-like phenotype with viral infection. So antivirals might actually look promising. And there was one study done a long time ago with antiretrovirals that seem to improve or rather ward off development of Alzheimer's. So we're looking again at the infectious link with Alzheimer's, as well as the just accumulation of stuff with age. Okay,
Very
so
good.
hopefully there'll be some good news in the next couple of years on the Alzheimer's front.
Let's hope. Let's hope. Too many people, such a cruel disease.
Yeah, absolutely.
So I was looking at the list and I recognized some of the names. We've had people
So do
tune
I.
all over the globe and all over the country and from all different age groups and alumni from different generations. So thank you all for being with us. And Lynn, thank you for an amazing talk today. On behalf
Thank you very much. It's lovely to see you all. Thank you. Okay, bye-bye.
Bye-bye.
Bye.